In the last video, we explored how individual amino acids link together via peptide bonds to ultimately form strings of covalently bonded amino acids, which fold up to yield protein structures, such as the schematic that we see here in the background, which represents the three-dimensional structure of a complex protein. These three-dimensional structures of proteins are critical to the function of proteins. Without the specific correct three-dimensional structure, disease will result. And there's a variety of diseases that result from disorders in protein folding. One prominent example of that is a variety of types of dementia, including Alzheimer's disease. By the end of this video, you will have a better understanding of the different levels of organization of protein structures so that we can interpret information about structural diagrams such as these. Generally, proteins are broken into 4 levels of organization, starting with the so-called primary structure, then at the more complex level, secondary structure, tertiary, and then finally quaternary as the most complex, highest level organization of proteins. Let's talk about what we mean with each of these different structural levels. The primary structure, shown here in a schematic where each individual amino acid is represented by a circle, refers to just the sequence of amino acids listed from N-terminus to C-terminus if we're listing them from left to right. So the primary structure, in other words, actually gives us no information whatsoever about the three-dimensional shape of the protein. It just gives us information on what the sequence of amino acids are so that we know if alanine is at the N-terminus of the protein, we would know that the sequence is alanine, tryptophan, phenylalanine, glycine, and so on and so forth. So it tells us what the order of the individual amino acids are listed from amino or N-terminus to the C-terminus, also known as the carboxy terminus. In order to understand how those amino acids are arranged together into a three-dimensional shape, we jump to the next level of organization, which is referred to as the secondary structure. With the secondary structure, what we're referring to are a couple of different repetitive structural elements that are very common across different proteins. Through hydrogen bonding, proteins commonly organize into so-called alpha helical structures, like this, where hydrogen bonding amongst the strands here helps hold this together into a helical shape. Another common secondary structure, these structures called beta sheets, where we have, shown with the arrow here, a sheet or a flat accordion type structure that lays flat and then coils back to form another sheet and possibly another sheet and another as we go through. With beta sheets, we can take a closer look at how these are laid out. When we have a so-called beta sheet, what we'll observe is that as we look at the strand of amino acid, which you can trace through here, the R groups hanging off here as the side chains, and we can recognize the peptide linkages here, where we have a carbonyl directly bonded to a nitrogen, here another carbonyl directly bonded to the nitrogen, so on and so forth. And what we recognize as we go through is an array of hydrogen bonds. Here, for example, where there's a dot, dot, dot to represent that hydrogen bond, here, 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 and so on. And this network of hydrogen bonding that we see along the backbone of this sheet will provide the structural basis for why the atoms organize in the way that they do to give this overall pattern that we refer to as a beta sheet, which we signify with these arrows present in the abbreviated overall structures of the protein. But if we looked more closely at what's represented by that arrow, it's actually this series of hydrogen bonds that allow us to have 2 strands of amino acids that are associated with one another through all of these hydrogen bonds to organize into a very specific so-called secondary structure. So at the secondary structural level, what you need to be aware of is falling into that category are alpha helices and beta sheets, like the beta sheet that we see down here. These are our partial levels of organization. They're referring to the structure of subsets of the overall structure of the protein. So within a huge protein structure, part of that will be organized as a beta sheet in many cases, and other parts will be organized as alpha helices. Then when we jump to the level of the so-called tertiary structure, shown here as a schematic, that refers to the complete three-dimensional structure or three-dimensional shape of an entire peptide chain. So that's why we show here, if we take a look at this, this represents one chain because we start our chain right here We follow that snake through, all the way through, finally ending over here, kind of eclipsed behind that other chain. And this is all folded up into a three-dimensional structure. So tertiary structure, the bottom line is, that represents the three-dimensional shape of a single peptide chain. So all of the amino acids in this tertiary structure are covalently bonded to one another. In many cases, tertiary structures will also have metals in association with them, such as iron, zinc, cobalt, and a variety of others. And those metals are essential in allowing the protein to form its three-dimensional shape or its tertiary structure. So it's quite possible in tertiary structure that there will be ions present as well. And that is why a variety of minerals are essential to life and why you can buy a variety of minerals such as iron supplements in the grocery store and elsewhere, because those are required for the proper three-dimensional tertiary structure of the entire protein. When we change one amino acid within the entire chain, sometimes a massive change to the three-dimensional structure can result. And that's pretty wild if you think about it, because what that means is if we mutate or change one amino acid out of the entire protein, sometimes the protein will change three-dimensional structures and no longer be effective as an enzyme. And this is true of a variety of genetic disorders where changing just one or a few amino acids in a particular protein results in the development of a disease because the protein can no longer function correctly. Finally, we look at the quaternary level of organization of proteins. And what this refers to is the association between multiple separate peptide chains. So in other words, in some cases, multiple separate proteins come together. They're not covalently bonded, but instead, through electrostatic interactions or hydrogen bonding, they affiliate with one another closely and work collaboratively, being bundled together into a single large three-dimensional structure. We show this here in that when we look at this tertiary structure, which is gray and pink, we see that tertiary structure showing up here in the upper right-hand corner of our quaternary structure and down here in the lower left-hand corner of the quaternary structure. We also see this brighter pink protein bundled up here. And we're seeing here in the quaternary structure the association of the gray protein with the pink protein to ultimately represent two different proteins that are closely associating with one another through intermolecular interactions in order to form this quaternary structure. And some proteins do indeed require a quaternary structure in order to be effective. Here we now take a closer look at an example tertiary structure of a protein. We recognize this as a tertiary structure because we see in this structure both beta sheets, shown in blue, represented by these arrows, as well as these old school phone cords, which are alpha helical shape. We also see regions of less organization in the structure, shown as these gray or black dotted lines, which represent what we call random coils, or regions that are not as heavily organized and ordered as beta sheets and alpha helices. Now, when we think about how proteins organize into tertiary structures, we think of some of the same types of interactions that we have looked at previously. Those are hydrogen bonds between specific R groups or the backbone of the protein, such as hydrogen bonds between the nitrogen and carbonyl groups of amides with hydrogens through hydrogen bonding. We look at electrostatic interactions in cases of R groups that have positive or negative formal charges. We can look at van der Waals forces when we are evaluating the interaction of nonpolar groups with one another. So these types of intermolecular interactions that we have focused on previously will work here to organize proteins into the necessary tertiary structures to allow them to have functions. Another type of phenomenon that happens within proteins to help organize them into tertiary structures is many proteins have multiple thiol groups. Thiol groups were SH groups. SH groups are the R groups, the variable group that you find on a cysteine amino acid residue. And what happens with those cysteine residues is that back in our thiol unit, we learned that SH can be oxidized to form disulfide groups, where we have a sulfur-sulfur bond. And indeed, in proteins, when two thiol groups are properly oriented, such as here in this schematic where I'm highlighting, and here, if they come into close proximity with one another, they can be readily oxidized to form a disulfide bond that helps link together those two portions of the peptide chain through a covalent bond here and can help drive the tertiary structure, the overall three-dimensional structure of the protein. And here in this particular protein, we see lots and lots of disulfide bonds represented by these red dotted lines here. And keep in mind the disulfide bond is a covalent bond because the two sulfur atoms are linked through a covalent bond. It is not an electrostatic interaction or a hydrogen bond-like phenomenon. It's literally a covalent bond. So now that we've looked at this information, what should you be able to do with it to apply it? You should now be able to take a look at the schematic of a protein and pick out key secondary structural elements such as alpha helices and beta sheets. You should recognize what we're referring to when we talk about a tertiary structure of a protein and be able to distinguish that from a quaternary structure, a secondary structure, and a primary structure.